Well, thank you very much for inviting me to speak today. I'm a bit of a fish out of water now because, as you've heard from the introduction, I'm from the pharmaceutical industry, and I have been in that industry for most of my career, the last almost 20 years. But very recently, I've joined this company, Bioleap, about 2 years ago, because it has a very novel approach to designing novel molecules. And as you'll understand, especially in the pharmaceutical industry, the only thing of value is novel molecules, molecules with intellectual property protection so that we can sell prescription drugs protected by patents. So I should say that the reason I joined Bioleap is that I felt that the pharmaceutical industry, and you'll hear in a few moments, had reached sort of a plateau in its ability to discover novel molecules. And they needed a change of some sort. And I think that the Bioleap, as you'll hear, has one very interesting technology to help us improve that early stage discovery. And I like to say that the whole purpose of our company is to generate that new intellectual property in a different way than it's been done traditionally. Okay, so with that, I should also mention that the reason I'm here is because I think this type of technology can be extremely applicable to discovery in the cosmetics industry as well. And quite recently, we've just signed an agreement, a collaborative agreement with Unilever to, in fact, apply our technology to consumer product development in the aging area. Okay. So next. Right, so a little bit of history in the pharmaceutical business. It's a pretty old business, as you'll know. Started probably in the early 1900s. And first sort of came into prominence with the discovery of novel antibiotics in the 1930s and 1940s. But over the next 50 years or so, until the early 1980s, pretty much discovery in pharmaceutical was pretty random. And the idea was basically take some compounds from natural sources or synthetic compounds and sprinkle them on your test object, whether it's cells or animals or whatever it's going to be, and then look for some sort of positive effect. A lot of this involved a lot of hoping because you didn't know what was going to happen. And oftentimes, even if you had some effect, you then knew nothing about how the compound actually worked. You had some sort of positive result that you had some effect, positive of some sort. The next stage, once you had that initial early lead compound with some modicum of some efficacy, was to then try to improve the potency of the compound in that test system, usually an animal model, and then improve the pharmaceutical properties so you could actually make it safe enough to put into human beings. And this was largely done through, again, a hunt and peck medicinal chemistry, is what I call it, sort of feeling your way through the dark with only having an assay, a biological test as your guide. So nothing more than that in terms of your ability to think in advance of what you'd like to make. And in the chemistry circles, this sort of chemical modification, small changes in the chemistry of the molecule, was referred to as basically methyl ethyl butyl, the idea that you're making random substitutions until you finally hit something good. All right? And oftentimes it took a lot of compounds to make, upwards of thousands of molecules before you'd find something at all. So that was until the 1980s, and by that point, biology was starting to now advance to the stage where we started to understand some of the biology behind various disease processes. And in the 1980s, a pretty dramatic shift occurred from going this sort of empirical discovery to a very target-based discovery. So we're trying to understand the disease biology and tie the disease pathology to a particular molecular target. That was the breakthrough of the 1980s, and then with that, of course, the discovery process, as you can see on this slide, became quite a bit more complicated. But this is now the paradigm that's been used for the last 30 years or so in the pharmaceutical industry. And it basically comes from the idea that there's some particular molecular target involved in a disease, diabetes, for example. There's a number of different molecular targets, enzymes and receptors involved in that particular disease. And once you identify which ones are going awry, you can then have a molecule designed to that particular molecular target that would change the biology and help you treat the disease. And so it involves understanding the protein structure of the actual target, and then a lot of biology and chemistry in this phase to actually find inhibitors or activators of that particular molecular target. Now, one thing you're going to hear a lot about today is this phase right over here. So once you've identified what the actual molecular target you're interested in is, you've then got to find what's called a lead molecule, something that actually has some effect against that particular target. And over here, this is called high throughput screening. This is, again, the existing paradigm in big pharma. The idea is that you take that molecular target and you throw literally millions, and often between 1 and 10 million compounds at that one target, looking for some sort of early stage indication there's some sort of efficacy. This, again, is, tantamount to basically gambling. You're taking lots and lots of tries, and very rarely, actually, do you actually come up with very good compounds here. Assuming you can come up with something there, then you get into the lead optimization phase, where you actually use medicinal chemistry, again, guided by nothing more than an empirical assay, again, feeling your way through the dark, chemically speaking, till you get to a compound worth putting into man. This whole process right now takes on the average of 12 years from the time you identify the target. It costs about $1.5 billion US to actually have a launch product. So, long, expensive, and not very rational, at least at this stage. Okay, so Bioleap is one of the things where our company actually operates, is in this phase, trying to cut out a lot of what we would refer to as unproductive work. Okay, why is this not going? There we go. All right, so a little bit about high throughput screening. As I mentioned, this is one of the things I'm going to talk a lot about. The idea is, and I've already said it largely, is it's robotic-driven testing of compounds. Again, between 1 and 2 million typically, but up to 10 million oftentimes. Right. Now I'd like to tell you a little story about sort of what's gone wrong in big pharma in terms of using this paradigm I've already described. So now I'm talking about the early phase of discovery here. This is before we've got a clinical candidate, right? And this was the experience that we had at GlaxoSmithKline about a decade and a half ago. And it started with the idea of trying to find a new antibacterial, a new antibiotic. At the time, GSK was looking for a replacement to Augmentin, which was a broad-spectrum antibiotic used in the community. And the compound was going to be going off patent, and they needed a replacement rather quickly. So they started in 1995 a huge program to discover a new antibiotic. And it was based on the idea of genomics. I don't know if you folks know about genomics, but this was the idea that you could identify all the genes in a bacterium and determine which ones are important for survival. And then once you've determined which ones are important for survival, you then try to find inhibitors of those molecules, right? That's the whole idea. And so what GSK did is they said, well, look, we're going to use this genomics approach, and we're going to come up with a bunch of potential targets, 160 of them, actually. 160 targets, molecular targets, all of which look pretty good. They did these high throughput screens, which I just mentioned, 1 million compounds per screen. So you talk about 70 million compounds were looked at over 7 years. And very few, what we call hits here, promising starts. And at the end of 7 years of work, $250 million, probably 200 people over that same course of time, not a single candidate worth putting into humans. Not one. So our response to this, of course, is there's got to be a better way than this. This is basically banging your head against the wall and hoping to come up with something better. It just didn't happen. So this sort of lack of success has led to, over the past decade or so in the pharmaceutical industry, to very dire circumstances. The pharmaceutical industry is in actually pretty deep trouble right now, and if you're looking at it at all from the outside, you're watching companies merge quite a bit. And the reason why is pretty much explained on this side. First and most importantly is that a lot of the blockbusters of the 1990s and early 2000s that were selling billions of dollars per drug are now going off patent. And when a drug goes off patent, it typically loses about 95% of its value in the first year. So if you had a $2 billion a year drug this year, and next year goes off patent, you've got about a $200 million drug. So for example, Pfizer is about to lose patent protection on its biggest drug, Lipitor, which makes them $12.5 billion a year, and it's expected to go down to about $100 million a year after that. And all the generics companies on the planet are lining up to make knockoffs of that particular compound. So you can understand why companies really have to work very hard to replace those big, big value products. Pfizer, no one knows what's going to happen to Pfizer next year, quite frankly. When you lose $12.5 billion and you're only making about $35 billion, That's a big chunk of your entire revenue. Okay, so that's one big thing. Lost patents, and then the problem is compounded by a very poor productivity in terms of new drug development over the past 15 years. I'm showing this on this slide. Basically what I'm showing here is the number of drugs in red and the amount of it's costing to make those drugs. Well, we're getting fewer drugs for more money. That's not good. So research productivity is the main concern right now in pharma. How do you discover new drugs? How do you get at them cheaper, better, faster? Because what we've been doing for the last decade and a half has not been working. Okay. So Bioleap's answer is at least work in the very first part of that particular pipeline in the early discovery phase. And we're trying to change the paradigm from what we consider gambling, throwing random compounds and hoping you get a hit, to, if you will, design, architectural design. We are going to design molecules specifically for a particular target. It is a hypothesis-driven process, not an empirical process. And so what do we do? Well, as I said before, we stay in the preclinical space, so we're only interested in identifying those early molecules. We'll let somebody else bring them into the clinic and further develop them. But this is a big issue. I mean, you can't find good lead compounds. As that example for the new antibiotic discovery I just told you about. Very hard to find those initial starting points. We are a computational chemistry company, so we don't have no laboratories. We only do design work, and it's based on an understanding of the molecular target at the atomic level, essentially. What this methodology allows us to do, as you'll see, is gain access to much more chemical diversity. We can look at just broader types of chemistry in general. One of the things you'll see, I think, was a problem with the failures in the recent past. And more importantly, is once we look at those molecules, we can tell you, before you actually have to make them, how well they're going to bind to their molecular target. So in essence, what this means is we're not going to make a lot of silly molecules. We're going to design a molecule, we're going to figure out how well it binds to its particular target, and if it binds well, we'll make it synthetically, and if it doesn't bind well, or predicted to bind well, we're not going to make it. That's going to cut down the amount of resources necessary by, we estimate, about a factor of 10. So I'll again refer to Darius' talk yesterday, this idea of becoming greener and more sustainable. Instead of making thousands of molecules to come up with your clinical candidate, we hope to make hundreds. And that saves all sorts of not only effort on the human side, but many of the work that you have to do drug discovery involves radioactive materials, chemical solvents, all various nasty things for the environment. And the idea, by cutting that entire process down by a factor of 10, we would be a much greener and, again, sustainable process. Okay, so some of the key problems, I've alluded to some of them already, but just to reiterate, one, hard to find that initial first compound, right? Because you're looking for a lot of different things in that high throughput screen. And here's a cartoon, really, of what you're doing. You have a molecular target, which I've been alluding to quite a bit, and it has some little pocket here you're going to want to interfere with. This pocket is essential for the normal functioning of that particular protein. And in most disease processes, you want to inhibit that. You want to block it. So what you do is you get 1 billion compounds, or 1 million compounds, whatever number of compounds, and you screen them literally one by one until you get to something that, again, from a random set of compounds, might fit. And that, once it binds there tightly, now you've inhibited the product, and that's a lead compound. So as you can imagine, again, total gambling. Let's assume you've even found that molecule now. Next thing is you've got to now change that molecule, because it's not perfect when you first find it. You've got to change it so it's actually useful in humans. It's got to be safe. It's got to have the right pharmacokinetics to be systemically available. It's got to have a million different things. And so that is the process that's called lead optimization. And this is very difficult, because you've got to hold all the good bits about the molecule constant when you're modifying the chemical structure, and then change the one thing you want to improve. So it's not an easy thing to do. And again, equivalence is essentially feeling your way through the dark room, because the only thing you've got to guide you is the empirical test of the molecule you've made. So the process is, chemist makes a molecule, he has some idea about why it might be better, he tests it, and then he says, better or worse, sort of like getting eyeglasses, right? And if it's worse, you throw it away, and then you try again. And that can take a lot of effort. So the chemical diversity thing, this is the first issue I was alluding to. You really need access to a lot of it. And the example I'm showing here now is, this is a plot of, we call it chemical space, right? Sort of a visual representation of the different types of molecules out there. And what I'm showing on this particular plot is the molecular weight, how big it is, and how greasy it is, essentially. And what I'm showing here is that when we did those screens for the new antibiotic at GSK, that first example I showed you, we screened through a typical corporate collection of molecules. Well, it turns out the most high-throughput screening libraries, the compounds you're going to use to look at, occupy a part of chemical space indicated in these blue diamonds. Right? So they're all localized to one particular area. That just happens to be the nature of compounds in libraries. Well, it turns out the most marketed antibiotics, the types of things we were looking for, they're down here. So you might say that we were just looking in the wrong place. We screened millions of compounds, and we were looking through a set of molecules over here, and we needed to look to a set of compounds over here at different types of this chemical space. Right? So we were looking in the wrong place. Now, if you had a methodology that you could look more generally around all of that chemical space, you'd have a better chance of finding your molecules. And essentially, that's what Bioleap is going to allow us to do. Where we can look in all these different chemical spaces. Most high-throughput screening libraries, corporate or otherwise, are localized in one very small spot. They just can't make the types of compounds out here. So you're never going to get to look at everything you need to look at. And what typically people say in high-throughput screening is you've got to add more compounds. We've looked at 1 million compounds today. Well, we need to look at 10 million. Well, this is what we say. Adding more hay does not help you find the needle, right? It's got to be you got to clear away the hay and get the right type of hay to look through, as it were. Okay. So what do? Bioleap, what we do is a little bit different approach, right? So we're going to do what's called fragment-based ligand design. And instead of looking for a molecule all at once, as you do in high-throughput screening, right, we're going to look for only little bits of that molecule, parts of it, potentially, and we're going to be focusing in on very small parts of the actual target protein. So we would argue that's a little bit easier problem to solve than a high-throughput screening problem. I've sort of diagrammed that on this slide. In the top panel here is an example of what you have to do in high-throughput screening. Right? You've got your molecular target in this yellow thing here, right? And you're looking for lots of compounds to fit into that perfectly. Well, most compounds don't do that because there's just too many different things you've got to accommodate. You've got to be right on just too many different ways. Whereas if you could break the problem down, essentially what fragment-based design is, you're looking at the same receptor site down here, but now we're only looking for compounds that bind here, and then compounds that only bind in that little small bit, and then only compounds, and then we can link them together at the end. So this is building the molecule up bit by bit rather than looking for the home run all at once. All right, so that's the essence of what we're going to be doing. Now, we can do this either experimentally or we can do it computationally, and we do it computationally. Now, let me show you how this can work. So what I'm showing here now is, and the title says it all, right, that this is a very tight binding drug. It binds very well to its molecular target. It's a beta-lactamase inhibitor. And basically, it's comprised of these 3 chemical fragments. This is how this molecule is designed. Right? These 3 fragments individually have very, very low affinity for the target. They don't bind very well at all, in fact. Okay. Now, how do we start looking at that comment? How do we look at that? Start to look at the chemical space, more of that chemical diversity? Well, you can imagine that if you were screening in the way I was telling you before, and you were varying this position by 1,000 different potential fragments, right? If you could look at not only this fragment, but 1,000 other different fragments in the same position of that molecule, well, that would be good because you now have 1,000 compounds. Well, now let's vary this one in 1,000 ways too. You multiply the 2 together, now you're up at 1 million. Now the third position, we vary that 1,000 ways. Now you're at 1 billion compounds you're capable of looking at. We're talking about 3 orders of magnitude now more, and this is a very simple example, than you could possibly look at in a high-throughput screen, doing it the old-fashioned way. The access to different chemistries here is just phenomenally different and hugely larger. All right. So what did, again, so let me tell you about how we actually go about doing that process. And since we're doing this computationally, all in silico, we're not doing this experimentally, our process is as follows. We have software, calculations, right, that run And then we have tools. These are on-screen tools that allow chemists to manipulate these data. I'm going to show you what these data are in a second. And in the very middle here, you actually have an old-fashioned medicinal chemist who his only job is to get ideas from all these data, because he's a very well-trained chemist that knows how to put molecules together. But he needs ideas of what to put together. And so what our platform is going to show this chemist is, here are all the possibilities on the target of what we already know binds to that target. So you're working with a stacked deck, as it were, right? Instead of randomly trying to find something that binds to the target, which is usually how chemists work, we're giving them a set of things we already know bind. Now, just put them together properly is all we're asking. And so how do we do that? Now, this is a little bit of thermodynamics here, but I'm going to go through it very quickly. The point of this slide really is to show you what the output of the calculations are. So basically, you have to start with a three-dimensional structure. That's in a blue ribbon here. This is p38 kinase, a very standard target in pharma. And what we're trying to do here is find out where a single fragment, and here I'm showing this is the naphthalene group, where does it bind on that target? So we do a lot of calculations that are thermodynamically rigorous, and I won't go into it here. But basically what it tells you is that you can find out where that fragment binds as a function of how tightly it binds. So we know where this fragment binds on this target. Some places it binds pretty loosely, and in some places it binds extremely tightly. But we know all of them, right? So this gives you a map, if you will, a map of where this fragment binds and with what relative binding affinity. Okay? That's what you got. That's one fragment. You can do this as many times as you want. We typically do 200 fragments, but you can do 1,000, you can do 20,000, you can do as many as you'd like, building up as much diversity as you want. The end result here is basically a fragment map of all those different fragments. And the example I usually give, the way to think about this, is it's a Christmas tree, if you will, and it's got ornaments on it. And you know where all those ornaments are and how tightly they're bound to the tree, how hard it is to pull them off. Okay? The question for the chemist is, how do you link up those ornaments to make drug-sized molecules? Right? That's shown in this slide. So this is the user, these are the user interface. This is the actual thing that chemists look at. And what does it tell you? Well, there's a couple things in this slide I'm going to tell you about. One is on validation of our technology, and I'll do that very briefly. And then how the chemists would use this information. So what I'm showing here in this ball and stick model is an actual ligand, a crystallographic ligand. This is actually experimental data of that structure I just showed you beforehand. Right? So if I go back a slide, this is the p38 kinase, and there's a ligand. That ball and stick model actually binds right there. Okay? So we know this is actually a real result. Now what I'm showing you here in these multicolored things, that is the calculation we just did for naphthalene, that fragment I showed you in the previous slide. And what I'm showing you is that it basically exactly overlaps what we know is already bound to the target. So by pure calculation, with nothing else except a calculation, we found a fragment of a known ligand in exactly the spot it was supposed to be. So what does that tell you? It tells you we can find fragments that bind to the target, and we can find where they bind precisely correctly, just by calculation. We could find this fragment if we actually looked for that one, and we could find this one. So we could, in principle, by linking these fragments together, have built this molecule from first principles without ever having done any actual experimental work. Now, so that's sort of a little bit of validation. Now, what does the chemist do? Well, what is the chemist can then look at this particular naphtha group in the middle here. He can literally click in a computer sense on any one of the atoms here that he might want to modify to start building this into a real compound. And when he clicks on that, he gets a pull-down list of all the different fragments that are nearby. So he can, this is a list of all the things he can modify that molecule with locally. So you just can add it in. And by the way, it tells you how much affinity you get when you add that in. He can check that atom and see what's out here. He can check this one, see what's out here, and just build it that way. All along the way, not making a single compound, but in fact, adding this as you go and calculating and predetermining how well the compound you're thinking about making will actually bind. Okay. So here's a cartoon to explain essentially what I just said there in those two more complicated slides. This is what I would say is that fragment map I described to you. So this is the Christmas tree with all the ornaments. Here's the little fragments. Here's the molecular protein target in the blue. And what a chemist now does is he looks at these various things and he says, oh, let's link these things up. and then he can calculate how well that particular molecule is going to bind. Well, you can imagine linking these things up in all sorts of different ways. Here's another example of linking them up. And here's another molecule, completely different. Chemical diversity thing again, right? We can calculate with this, how well this binds. And we can then determine whether this binds more tightly than this one, for example. And if it does, we'll only make that one. We're not going to make both of them. We're only going to make one. And you can now expand this to the logical extreme conclusion. What if you design 1,000 of these? How many are you going to make? Well, in the old days, you made all 1,000 because you had no way of evaluating whether they're going to be any good or not before you made them. Here, we make 1,000 of them, and we draw a line under the top 50. And that's all we're going to make. So we're cutting down the amount of work by 10 to 15-fold. Okay. And how good are we at doing this? So what I'm showing here is a plot, right? And what it is, it's a correlation plot between what we calculate the affinity to be and what the actual experimental data show, right, after you make the compound. So we did this two ways to make sure that we weren't cheating, right? We did literature values. These are known compounds that act against this particular target. And then we made some of our own that we predicted would bind with certain affinity. And what I'm showing here is that not only can we take literature compounds, break them up into fragments, use our technology to figure out how well they bind. These are in red. And of course, they now bind exactly as predicted. That's this linear correlation here. But we can also design new molecules. These are in blue. And lo and behold, they actually bind exactly as they're predicted to bind. So what is that telling you? It's telling you that we can tell you what the next best compound is to make. Because if we calculate the molecule is going to be here, we're not going to make it. Doesn't have enough affinity. And this line shows you that, since it's correlated with experimental data, is telling you that when we say it's going to bind well, it's going to bind well. I'm sure that's clear. Now, one of the things people often say is, well, how are you different from a lot of other computational chemistry methods? Well, in a short sentence is we account for the thermodynamics of binding better. And to be even more technical, we account for entropy. And what does that mean, actually, at the end of the day? What it means at the end of the day is that if you don't account for entropy, which just about every other computational method does, you get pretty much a scatter plot. So this is the data with us. This is the data with existing computational methods. This is not predictive, right? You can't be sure that the next molecule you're going to make is going to be as predicted. Look at that one right here. These two are basically right over each other. There's no difference there. Okay. And so how valid are we? Well, I think it's important to take a look at, again, some of the scientific literature. And there's a very well-known paper actually published by GlaxoSmithKline, their chemistry group, which basically concludes that all the computational methods out there existing they don't work. So none of the docking programs, these computational programs, can measure ligand binding affinity, can predict it. Now, they've published this, and of course we're saying just the opposite. So we went to GSK and we said to them, look, you published this paper, we actually think we can predict what the binding affinity is going to be. You've just said publicly that you can't do it. Right, so they gave us a blinded set of compounds, a bunch of compounds they knew the binding affinity for, but we didn't. And they said, okay, you rank order them. And so we did, and our plot was linear. They had just said in that previous paper, theirs was scatter plot. They could not predict it. We could. So this led to a deal within a couple weeks after they saw those data. So this is what's unique about our platform. The idea that we can circumvent a lot of random activity in chemical synthesis. We actually have a hypothesis-driven way of coming up with molecules that will bind tightly to the target. And that's really what you need to do. A lot of applications for this particular platform. I've listed a bunch of them here. One I'll just focus on, and that is we can control the physical chemical properties of the molecules, right? Because as we're building them up, fragment by fragment by fragment, we can introduce greasiness or hydrophilicity, whatever you'd like to, any sort of physical chemical property you'd like to do. What that means for the cosmetics industry, for example, is we can, assuming you guys know what kind of chemical properties you need for creams and topical types of applications in your molecules, we can engineer those fragments to be in the types of chemistry you need. So it's going to be very different than I use for pharma, for example. Pharma, I'm looking for systemically available drugs, things that you can actually take by pill and they will be systemically distributed. I think in cosmetics, largely you're looking for things that are creams or things that are topically applied. Well, those are very different things from a chemical perspective. You're going to want a very different molecule that goes inside your body compared to the one that you might want to spread around on your skin. But the platform here allows us to differentiate at the earliest stage about the types of chemistries we're looking at. Right? So we can literally tailor-make molecules for the types of things that cosmetic molecules need. This is only to say that we are not restricted by any sort of target class. So if you understand the molecular targets involved in whatever cosmetic indication you're looking for, we can probably work on that target. So for example, if there are molecular targets associated with aging, for example, wrinkling or age spots, and there are, once you've identified that target, we can then address it. And here's what I call the money slide. We've compared ourselves here directly to the typical process in the pharmaceutical industry anyway. So on the bottom here is a typical timeline and resource requirements for a pharmaceutical project to get to a clinical candidate. All right? So this is not the clinical trials. They would be way out here. This takes at least 4 years in pharma, usually 6, but I'm being generous here. And as you can see, lots of compounds synthesized, over 2,000 to get here. Right? And this is one by one, feeling your way through the dark. Most of these compounds, as you'll know, will not work. Right? They're a waste of time. Requires huge infrastructure. Do that high throughput screening I mentioned. There are buildings that cost hundreds of millions of dollars to get these sort of facilities in place. GSK has 3 of them. They cost almost $600 million between the 3 of them. It's a very expensive infrastructure to maintain this. Now contrast that with our approach, right? Because we're doing this, if you will, smart molecule approach, we're only going to make about 150 compounds on the way to that development candidate. We have no infrastructure. We're completely virtual and much faster because, again, we're not testing as many compounds. So we're going to be there a lot quicker. Okay? And this is my last slide, only to sort of highlight that our approach now is sort of been an evolution from the pharmaceutical discovery itself, right? We've gone through the high throughput screening modes and the sort of hunt and peck chemistry, and now we're trying to become much more rational about how we do our discovery. It's very important to do that because the costs, as I alluded to early on in the pharmaceutical industry, are now skyrocketing. It's just too expensive to make drugs. Big companies can no longer stay in business. There's got to be a different way. And I would argue that you want to stop being you want to stop being insane, right? Insanity was always described as doing the same thing over and over again and expecting a different result. And the pharmaceutical industry has been doing that for too long, and now I think it's time for a change. And I think the cosmetics industry, likewise, could possibly benefit from this approach earlier on. Don't make all the mistakes that pharma did and going down that road of random screening and high throughput screening. So that's all I have to say. Thank you very much for listening, and I'll take any questions you have.